Hi everyone, thank you for watching this video tutorial, and I hope you find the information useful. This video is going to go over the steps involved in reading and grading a Plasmodium fish smear that has been hybridized with the ID Fish Plasmodium PF-PV combo test kit. To begin, you must ensure that you have the proper setup. For this test, we use a 100x oil immersion objective. You should also make sure that you have an LED fluorescent unit with custom filters and that both of the power switches are turned on. You should check to see that the slider is set to the correct filter. Having it set to the blue filter will ensure that you will see a green excitation when looking for PF through the eyepieces. Having it set to the green filter will ensure that you will see an orange excitation when looking for PV through the eyepieces. When you are obtaining your samples, first ensure that they are no more than 2 years old. You can read a single smear slide or a double smear slide, whichever is more convenient. The smear should be made according to the instructions in the product insert included with the kit. The grading table for Plasmodium fish is as shown. It is the same grading scale that is used for Giemsa stain reading of thin smears. In order to call the smear negative, one must read a minimum of 300 fields and find no parasites. As seen here, a grading code of 1 would correspond to 1 to 2 parasites per 300 fields. A grading code of 2 would correspond to 1 to 9 parasites per 100 fields. A grading code of 3 would be 1 to 9 per 10 fields. A grading code of 4 would be 1 to 9 parasites per field. And a grading code of 5 would correspond to an extremely high positive, having more than 9 parasites per field. Now we will go over how to efficiently and effectively scan the microscope smear in order to maximize accuracy. Since a minimum of 300 fields are required for negative and low samples, the best way to achieve this is to begin at one side of the smear, reading down one column. With the blue filter in place, you will be looking for green parasites. Then change over to the green filter and read up the same column, looking for orange parasites. Shift the field over in one direction and read down a different column, again in green, and up that same column in orange. Repeat that 2 more times and it should be adequate to get a sufficient reading. Since one column represents approximately 75 fields on a normal-sized thin smear, reading 4 columns would be equal to approximately 300 fields. Make sure to record the results on a form like this. Here are some examples of what to look for in a positive sample. This is a typical ring on a PF positive slide. Notice how it appears in green, but not in orange. Here is an example of an artifact. As you can see, it does not have the typical morphology of a parasite, and it also appears in both colors, which is a fairly good way to double check that the signal is an artifact and not true signal. For this test, since a parasite can only be one species or the other, it would be impossible for a true parasite to excite under both filters. Here we have a good example of a Schizont stage Plasmodium parasite inside of a red blood cell. Unlike the last example, this parasite is only exciting in orange and not in green. Here is a more typical example of a ring-shaped PV parasite inside of a red blood cell. It is exciting in orange and not in green. Again, here is some footage of a fairly typical artifact. Take note of how the green color and the orange colors are more yellow than a true signal would be. And here we have another PV parasite inside of a red blood cell. Here we have a low positive PF sample. You can see these ring-shaped parasites that appear in green and not in orange. Here's a zoomed-in picture of a gametocyte showing up strongly in green. This is a nice example of a Plasmodium falciparum parasite in the gametocyte stage. Notice the typical banana-shaped morphology. Okay, well that wraps up this video tutorial. Please view our other tutorials for more examples of artifacts and tips on troubleshooting. If you happen to have time, there is also a brief 10-question customer feedback survey on our webpage that we would so appreciate if you completed. The feedback helps us a lot in figuring out how to serve your needs better. If you have any remaining questions, please do not hesitate to call or email us and we will do our very best to assist you. Thank you for watching.